हेलो दोस्तों, आप सभी का स्वागत है आपके अपने चैनल आयुर्वेदा बायोस्फीयर में। सो टुडे वी आर गोइंग टू डिस्कस द न्यूक्लियर रिसेप्टर. दिस इज आल्सो नोन एज इंट्रासेलुलर रिसेप्टर. सो जो भी स्टूडेंट्स यूजीसी नेट, सीएसआईआर नेट, आईआईटी जैम की तैयारी कर रहे हैं तो ये टॉपिक उनके लिए काफ़ी ज़्यादा इम्पोर्टेंट है और ये सारे एग्जाम्स में कहीं ना कहीं इससे जो है वो क्वेश्चंस जो है वो रहते ही हैं. So in this lecture, we will be discussing, you can see here, endocrinology, the nuclear receptor superfamily, and we will also see the structural and functional organization of all the nuclear receptors. In short form, we used to write it NRs, okay? And we will be discussing type 1 and type 2 nuclear receptors and also its mechanism of action. तो आइए शुरू करते हैं. सबसे पहले हम देखेंगे इंट्रोडक्शन और टू इंट्रासेलुलर रिसेप्टर विच इज आल्सो नोन एज न्यूक्लियर रिसेप्टर्स. सो व्हाट आर इंट्रासेलुलर रिसेप्टर्स? So intracellular receptors belong to a large superfamily of ligand activated intracellular transcription factor. So these nuclear receptors or intracellular receptors are ligand activated. It means some specific molecule called as ligand are necessary to activate these intracellular receptors. These are basically transcription factors. What are transcription factors? They bind to the promoter sequence of the DNA and there they used to either upregulate or downregulate the gene expression. So either it can upregulate or it can downregulate the expression of several genes present on the genome. These are soluble proteins that can bind to specific DNA regulatory elements, and these specific regulatory elements are known as response elements. In short form, it can be written as REs, and act as cell type and promoter specific regulator of the transcription. So basically, ligand regulated intracellular receptor. It means ligand is necessary to activate these intracellular receptor, and these are basically transcription factor which helps in gene regulation. So it is one of the important feature of intracellular receptor, gene regulation. Okay. Now let's see the next point. In contrast to other transcription factors, so what is unique about these intracellular receptor? So the activity of intracellular receptor can be modulated by binding to the corresponding ligands. As I have told before that these nuclear receptors or intracellular receptors are ligand activated receptors. So what are ligands? Basically these are small lipophilic molecules. Means these are hydrophobic molecules. Hydrophobic molecules, or you can say lipophilic molecule. So why they are lipophilic molecules? Because they have to penetrate the biological membrane, because they have to cross the nuclear membrane. Suppose this is nucleus and inside the nucleus there is DNA, and in that DNA there is presence of response element where the receptor has to bind. So ligand, suppose L is ligand, it has to cross these nuclear membrane. So this is the nuclear membrane, and any membrane is made up of, you can see, this is phospholipid bilayer where most of the portion is TEL, that is lipophilic. That's why you can see ligands are lipophilic molecules, so that it can easily cross the nuclear membrane of the nucleus and it can bind to the response element and activate this particular receptor and following the gene expression or regulate the gene expression. Now next point is intracellular receptors may be classified either according to activation mechanism. So according to activation mechanism, these are of two types, type 1 and type 2 nuclear receptor. So we will see these types in detail in further slides, in upcoming slides. Okay. On the basis of sequence homology, these nuclear receptors are further classified into subfamilies 0 to 6. So we will see some examples of these nuclear receptors in further slides. Okay, so next point is in the non-liganded state. It means in the absence of ligand. If ligand is absent, then what happens? So these receptors reside in the cytoplasm. So it reside in the cytoplasm when there is no ligand. Okay, or it can reside in nucleus also in the absence of the ligand. But when they bind to the ligand, here hormone has been used in this place of ligand. Okay, so hormone is nothing but a ligand. When we talk about SHR, SHR is steroid hormone receptor. So for steroid hormone receptor, hormone is hormone is acting like a ligand. So hormone is nothing but a ligand for steroid hormone receptor. For example, estrogen, progesterone, testosterone. These are receptors where you can see hormone acts as a ligand. Okay. So in non-liganded state, these receptors reside in the cytoplasm or in the nucleus, and then upon binding to the ligand, means when hormone binds to these steroidal hormone receptors, these receptors undergoes dimerization either with the same receptor, then it will be called as homodimerization, or it can dimerize with some other heterodimeric partner, that is called heterodimerization. So basically they will dimerize. So it is also one important event, you can see dimerization. Okay, so after dimerization, they regulate either positively or negatively the gene expression. It depends upon the recruitment of co-regulatory elements. Recruitment of co-regulatory elements. Co-regulatory elements are basically of two types. One is called co-activator, which can bind to positively regulate the gene expression, and the other one is co-repressor. Upon binding to co-repressor, there is negative gene expression. So you can see it regulate either positively, okay, either positively or negatively. So it depends on the recruitment of co-regulatory element like co-activator and co-repressor. Okay, gene expression by binding to specific sequences in the chromatin. So chromatin also plays an important role in positive and negative gene regulation, termed as hormone response element. So these elements are present on the DNA where the hormone or the ligand binds. So these things we will see in detail in further slides. These receptors are evolutionarily conserved proteins that control a broad array of genetic programs, in turn may be responsible for embryonic development, reproduction, immune functions, and metabolic homeostasis. So you can see these are very important functions like embryonic development, reproduction, immune functions. So these receptors should be conserved in mammals, in human beings, so that at least changes can occur. So in upcoming slides, we will see the structure that is very common in most of the nuclear receptor. It means it has not been changed very much during the evolution. Why? The reason is because it is involved in most of the important functions like embryonic development, reproduction, immune function, etc. So this slide talks about structural and functional organization. Okay, let's erase this. Okay, yeah. So structural and functional organization of nuclear receptor. So you can see this is the 2D structure of nuclear receptor. So you can see the two ends. First end is N terminal and the another end is C terminal. And between the two ends, you can see 1, 2, 3, 4, and 5. These 5 domains are present. These 5 domains are present between the N terminal and C terminal. The first domain is N terminal domain, NTD. This is also called as AB region, or it is also known as amino terminal domain. And this region incorporates one or more autonomous transcription activation function 1. And these activation function 1 works in a ligand independent manner. Ligand independent manner, it means this domain is this domain doesn't require ligand to bind so that to activate the receptor. Means without ligand also the receptor can be activated. So this domain has the activation function 1, and the receptor which contain this activation function 1, they can work as in a ligand independent manner. But the exception in case of steroid hormone receptor. I have elaborated this in the first slide also. For steroid hormone receptor, these are ligand activated. So here ligand is necessary to activate the steroid hormone receptors like testosterone, estrogen, etc. So ligand here is hormone. So hormone is necessary to activate these steroid hormone receptors. That's why it lacks activation function 1. Instead of activation function 1, it has activation function 2, which acts as ligand dependent manner. Okay, so the next domain is DNA binding domain. So DNA binding domain, you can see this region is called as C region. Okay, so it is the most conserved in different families and some families of the nuclear receptor. It is responsible for sequence-specific DNA recognition because the ligand has to recognize the receptor which is bind to the specific sequence of the DNA. So the sequence should be specific. For that is DNA binding domain. Okay, so DNA binding domain is comprised of two zinc finger motif. You can see here two zinc finger motif is present here. Okay, so these two zinc finger motifs are also essential in some functions, like DNA binding domain has sequence elements termed as PDTA boxes. Okay, so P box is necessary for the specificity of the response element that is present on the DNA, and D box acts as an interface for dimerization. It means DNA binding domain has role in dimerization also, and dimerization is one of the important step in the process of gene regulation. T box, an interface for dimerization, it also helps in dimerization. And lastly, the A box, this is the DNA core recognition sequence. DNA binding domain in most of the nuclear receptor contains two types of signal. One is NLS, this is also called as nuclear localization signal, and the another one is nuclear export signal. As the name suggests, you can see for the entry, NLS signal is important, and for the exit, NES, it means export signal is important. So it plays a role in न्यूक्लियोसाइटोप्लाज्मिक सेटलिंग, यानी कि कोई भी चीज न्यूक्लियस में जा रहा है, न्यूक्लियस से साइटोप्लाज्म में आ रहा है, इसके लिए NLS and NES है. Okay, now the next domain. So let's take the next domain, that is the hinge region, region D. So hinge region, as the name suggests, you can see it connects the two domains, DNA binding domain and ligand binding domain. So hinge region basically a connecting link between DNA binding domain and ligand binding domain. So it acts as a hinge between C and E domains. The region confers conformational flexibility. So it gives conformational flexibility to the receptor. Region D further contains the nuclear localization signal or nuclear export signal as like the DNA binding domain. So it also contributes nucleocytoplasmic settling of the receptors. Okay. Now let's talk about the next domain, that is the ligand binding domain. So ligand binding domain is called as region E, and this is one of the most important domain of the nuclear receptor. It is called as hallmark of nuclear receptor. It is highly structured, translates a wealth of distinct physiological functions, mostly operated in ligand dependent manner. See, ligand dependent manner, whereas N-terminal domain has activation function 1, which act as ligand independent manner. Ligand independent manner. But this ligand binding domain, you can see it has activation function 2. It means it acts as ligand dependent manner. It acts as ligand dependent manner. So for steroid hormone receptor, it contains activation function 2. That's why we have studied that SHR is exception for N-terminal domain because it lacks activation function 1. Okay. Now, this ligand binding domain moderately conserved in sequence but highly conserved in its structure. So when we will see the structure of ligand binding domain, so it is mostly conserved in most of the nuclear receptors. It can be regarded as the second most conserved region after the DNA binding domain. Also, it acts as a binding site for different co-activator and co-repressor. I have discussed about this co-activator and co-repressor. These are called as co-regulatory elements. And what are the functions of co-regulatory elements? If you can remember, if you can recall, I have explained this, that these co-regulatory elements determines whether the expression is positively regulated or negatively regulated. When it will be positively regulated, when there will be co-activator recruitment. If there will be co-repressor recruitment, then there will be negative gene regulation. Okay. It also contains a ligand dependent activation function 2. I have explained it. And it acts as a major dimerization interface. See, dimerization is the main function of ligand binding domain. Whereas DNA binding domain has also role in dimerization, but the most important role of ligand binding domain is the dimerization. So this is the function of LBD, and often has a repression function because it can recruit co-repressor. Among all the domains of the nuclear receptor, LBD contributes most of the receptor dimerization process that I have discussed now. Okay, now we will see the carboxy terminal. Okay, so carboxy terminal domain, this is also called F domain, and this is the last domain present nearby the ligand binding domain. Okay, so F domain, you can see it is a least evolutionary conservation. That's why most of the nuclear receptor lacks this F domain. The function of this C terminal sequence is still ambiguous. Some studies suggest that this region plays a part in co-activator recruitment to the ligand binding domain and also determines the specificity of the ligand binding domain because it is present nearby the ligand binding domain. It also fine-tunes the molecular events related to the transcriptional properties executed via the ligand binding domain or the whole receptor. So this is very optional domain which is not present in most of the nuclear receptor. That's why it is the least evolutionary conservation. तो ये तो हुआ structural, and साथ साथ हमने each domain of the nuclear receptor के functions भी देख लिए. Now let's move to the next slide. Okay, so this is the same thing that I have explained in the previous slide. These are the N terminal, this is the C terminal, and these are the important domains. F domain has not been shown here because this F domain was very optional and not present in most of the nuclear receptors. And now you can compare these different domains. Okay, so all the points I have already covered in previous slides. Okay, so this is only for a quick view for you guys. Okay, so now let's see this figure to understand. This is the 3D figure where you can see a nuclear receptor has been shown, where you can see different domains. See, this is the ligand binding domain shown in red color. This is the hinge region. This is N-terminal domain, the blue color part, and this yellow color part is DNA binding domain. That's why this domain is, you can see it is bound to the DNA and specific sequence present in the DNA called as response element. So this specificity is recognized by this DNA binding domain, this yellow region part. So in this way, the nuclear receptor attach to the DNA, and after attachment, the two important process occurs. If you can recall, I have explained this. The first process is dimerization, either homodimerization or heterodimerization, and the second important process is recruitment of co-regulatory elements. And what are co-regulatory elements? Either co-activator or co-repressor. If co-activator will bind, then the gene expression will occur, means there is positive gene expression. If co-repressor will bind, then there will be repression. It means the gene expression will be negative. Okay. So now let's move to the next slide. Now let's see the classification of nuclear receptor. So in the classification of nuclear receptor, we can see there are total 4 classes. So nuclear receptor superfamily members have been classified or grouped in several ways. However, the superfamily can be most conveniently divided into 4 subfamilies based on DNA binding properties and dimerization preferences. The first subfamily comprises of group 1 receptor, which mostly forms the homodimers. It includes various steroid hormone receptors like you can see glucocorticoid receptor, mineralocorticoid receptor, progesterone receptor, androgen receptor, estrogen receptor. These are steroid hormone receptors. So this is, you can see, class 1 steroid hormone receptor. These are the examples. You can see this is the dimerization. More accurately, we can say it is a homodimerization of ER alpha, and further the co-activator or co-repressor will bind, and this is the DNA binding domain, which helps in binding to the sequence response element present on the DNA, and then gene expression will occur, means the transcription and translation process will occur. Now the repeat that is present on the DNA on which the DNA binding domain of the given receptor bind is inverted repeats in case of class 1 receptor. So actually there are 3 type of repeats that we will see in class 2 nuclear receptor. So basically the members that function as heterodimers with the RXR. See, RXR is a common heterodimeric partner of most of the nuclear receptor. This forms the group 2 and group 3. Means group 2 and group 3 nuclear receptor forms heterodimers. And the heterodimeric partner RXR. RXR is an obligate heterodimeric partner of most of the nuclear receptor. So group 2 consists of receptors whose ligands have been identified, and group 3, 1 minute, yeah. Group 3 includes the orphan receptors. It means their ligand has not been discovered or identified yet. These receptors are called as orphan receptors. Okay. So let's see the class 2 RXR heterodimers. Means these receptors which comes under class 2, they form heterodimer with RXR. You can see here PPAR alpha is given here. It forms a heterodimerization with RXR. Then after co-activator or co-repressor recruitment, then further the gene regulation will occur. These are some more examples of class 2 receptors. Now these class 2 receptors can bind to direct repeat or inverted repeat or inverted repeat. Direct repeat is nothing, you can understand that the repeats of these sequences, basically the nucleotide sequences, their orientation is same and they are present on the same strand. The inverted repeats, they are present on the opposite strands. Suppose if one is 5' to 3', the next sequence will be present on 3' to 5', and their orientation will be also in opposite direction. Their orientation is in opposite direction. The inverted repeat, they will be present on the same strand, but their orientation will be opposite. Okay. So these are the repeats present on the DNA. Now class 3 homodimers, you can see RXR alpha. It can form homodimer with each other, and further the gene regulation can occur. These are some more examples of class 3 homodimers, and it binds with the direct repeat. Means the same orientation present on the same strand. Now class 4 is monomer. So this is called group 4, and this contains the ligands like melatonin, cholesterol derivatives, and some receptors which are also called as orphan receptors because their ligand has not been identified yet. So these are the classification of nuclear receptor. Now let's see what is the difference between intracytoplasmic or intranuclear receptors? Basically, they both are called as nuclear receptors, but there is some difference that we can see here. Okay, so the term intracytoplasmic and intranuclear receptor are often used interchangeably to describe the receptors located within the cell, because both are the receptors present within the cell. Some differences we can see. Okay, so let's talk about the location. So intracytoplasmic receptor are primarily found in the cytoplasm, because as the name suggests, intracytoplasmic, so they are present in the cytoplasm. But in case of intranuclear receptor, they are predominantly located in the nucleus of the cell, even in their inactive state. Okay, so upon activation, what happens? So in case of intracytoplasmic receptors, they are activated upon binding to their specific ligands, means they are ligand dependent. Often these ligands are lipophilic because they have to cross the membrane. But in case of nuclear receptors, these are activated by their ligands, which can directly enter the nucleus and bind to the receptors. So here you can see the ligand directly crosses the nuclear membrane because the receptor is present inside the nucleus. But in this case, since receptor is present in the cytoplasm, so the ligand of intracytoplasmic receptor has not required to cross the nuclear membrane. Okay, in further slide it will be more clear, because I will show you in a diagrammatic view through animation. Okay, so this, now let's talk about the translocation. So since the intracytoplasmic receptors are present in the cytoplasm, and ultimately it has to move into the nucleus for the gene expression, so translocation is required. So once activated, they translocate to the nucleus to exert their effect on gene expression. But in case of intranuclear receptor, it is already in the nucleus, so there is no need of translocation. So it directly bind to the specific DNA sequence and regulate the gene expression. So here the translocation is required because it is present in the cytoplasm. Here no translocation is required because it is already present in the nucleus. So these are the examples. Okay, so most of the steroid hormone receptors comes under the intracytoplasmic receptor, and thyroid hormone receptor, retinoic acid receptor known as RAR, these comes under intranuclear receptors. These are also termed as intracytoplasmic receptors are called as type 1, and intranuclear are called as type 2. Okay, so intracellular receptors are often referred to as nuclear receptors because they are primarily located within the nucleus of the cell. So let's see the intracellular receptor type 1 and type 2. So as I have already discussed, these are of two types, type 1 and type 2. Okay, so type 1 and type 2 intracellular receptors. So type 1 receptor is also called as intracellular cytoplasmic receptor, and so let's see the intracellular cytoplasmic receptor. So you can see ligand here. So the receptor of intracellular cytoplasmic receptor is present in the cytoplasm. So it is present in the cytoplasm. So the ligand comes and cross the membrane and bind to the receptor present in the cytoplasm. Then further, the translocation of this receptor ligand complex will occur. So you can see this translocation is required in case of type 1 or intracellular cytoplasmic receptor. So when it translocate, then further all the processes like dimerization and co-regulatory elements are recruitment occurs, and then further gene regulation take place. So you can visualize here that why the translocation is required, because ultimately it has to move inside the nucleus for gene regulation, and since it is present in the cytoplasm, so it has to translocate. But in case of intracellular nuclear receptor, you can see the receptor is already inside the nucleus, so there is no need of translocation. So ligand can directly cross the nuclear membrane, and it can directly bind to the receptor which is already present in the nucleus, and further the gene regulation occurs. So basically both has to come inside the nucleus to express the gene. That's why both are both can be termed as nuclear receptors. These are the examples of type 1 and type 2 receptors that I have already discussed. Now let's see the concept of agonist and antagonist. This is also an important section that you have to understand. See, this is the helix 12 structure function model. So basically, this is the domain called as ligand binding domain, where you can see a ligand binding pocket is present, and helix 12 is the part of ligand binding domain. What are agonist and what are antagonist, you have to understand. See, in case of any we can take example of suppose we can take example of thyroid receptor, TR or THR. So in case of THR, what is the natural ligand of the thyroid hormone receptor? So T3 is the natural ligand. So any drug molecule that will mimic the action of T3, that can be called as agonist of the thyroid hormone receptor, or any drug molecule that will act opposite or antagonistic to T3, that will be called as antagonist. Okay, so if you have to enhance the expression of any receptor, then what you have to do? You have to treat it with agonist. If you want to enhance, if you want to repress, then you can treat it with a drug comes under the category of antagonist. मीन्स एंटागोनिस्ट होगा तो क्या होगा? वो रिप्रेस करेगा. एगोनिस्ट आप दोगे तो क्या करेगा? वो मिमिक करेगा नेचुरल लाइगेंड को, तो वो इनहांस करेगा. तो सपोज एगोनिस्ट जो है वो बाइंड करता है. ओके, तो सपोज एगोनिस्ट मान लीजिए बाइंड किया. सो एगोनिस्ट यदि बाइंड करेगा तो क्या होगा? यहां पर कॉन्फॉर्मेशनल चेंजेस आती हैं हेलिक्स 12 में. तो ये जो हेलिक्स 12 है, यहां पर कॉन्फॉर्मेशनल चेंजेस आती हैं. ये एक्टिवेशन फंक्शन 2 जो कि लाइगेंड बाइंडिंग डोमेन में होता है, वो कहीं ना कहीं रिस्पॉन्सिबल होता है फॉर द बाइंडिंग ऑफ द लाइगेंड. और जैसे ही लाइगेंड की बाइंडिंग हो जाती है लाइगेंड बाइंडिंग पॉकेट में, क्यों हुई? क्योंकि एगोनिस्ट के आने से हेलिक्स 12 जो ऑलरेडी मास्क करके रखा था उस लाइगेंड बाइंडिंग पॉकेट को, जहां पर एक्टिवेशन फंक्शन 2 था, तो वो हेलिक्स 12 क्या हो गया? साइड हो गया, जिसके कारण ये पोर्शन क्या हो गई? अनमास्क, यानी कि एक्सपोज हो गया. एंड फर्दर यहां पर क्या हुई? को एक्टिवेटर की बाइंडिंग, और जैसे ही एक्टिवेटर आया, इट मींस यहां पर जीन एक्सप्रेशन जो है वो होगा, और कोई एगोनिस्ट है तो वो टारगेट जीन को क्या करेगा? बढ़ा देगा. तो जब भी हमें रिक्वायरमेंट होगी ओवर एक्सप्रेशन करने की किसी जीन को, तो हम एगोनिस्ट का यूज करेंगे. इसके जस्ट अपोजिट क्या होगा कि आप किसी एंटागोनिस्ट से जो है वो डाल रहे हो. एंटागोनिस्ट बिल्कुल मिमिक करता है एक लाइगेंड फ्री कंडीशन को. इट मींस द हेलिक्स 12 विल basically change its conformation, but when the either ligand will bind or ligand can't bind. So if in the absence of ligand, what will happen? Helix 12 will mask the activation function 2. So activation function 2 यहाँ पे क्या है? masked है ligand binding pocket में, तो ligand जो है वो bind नहीं हो पाएगा, ligand free condition रहेगी. सेकेंड केस क्या हो सकता है इनएक्टिव कॉन्फर्मेशन का? सेकेंड केस ये हो सकता है कि ligand bind तो किया, देखिए ligand bind तो कर पाया क्योंकि helix 12 ने अपने कन्फर्मेशन को चेंज किया, द सिमिलर केस ऑफ एक्टिव कन्फर्मेशन, बट इस ligand के बाइंडिंग से यहां पर जो रिक्रूटमेंट हुई है वो को रिप्रेसर की हुई है इंस्टेड ऑफ को एक्टिवेटर. तो यदि को रिप्रेसर आएगा तो यहां पर टारगेट जीन का एक्सप्रेशन क्या होगा? नहीं होगा, यानी कि डिक्रीज हो जाएगी. तो मान लीजिए कोई भी जीन का एक्सप्रेशन बढ़ गया है, जो सर्टेन कैंसर्स में आप देखने को मिलता है, तो उसके एक्सप्रेशन को मान लो हमको क्या करना है? घटाना है. तो आप ड्रग्स ऐसे देते हो जो क्या हो? एगोनिस्ट का सॉरी एंटागोनिस्ट का काम करे. किसी का एक्सप्रेशन कम हो गया है जिसके कारण डिजीज आ रही है, तो आप एगोनिस्ट ड्रग दे सकते हो. So this is the mechanism of action of agonist and antagonist. So you can see in this graph also, this is the basal activity or transcriptional activity of the receptor. That's why it is written neutral antagonist. It means यहाँ पर basal activity है, means 0. यहां से मान लीजिए बढ़ेगी तो क्या होगा? तो आप देख सकते हैं ये क्या है? फुल एगोनिस्ट. इट मींस मान लो कि ये 100% एक्टिविटी को बढ़ा देता है. यदि 50% बढ़ रही है तो वो क्या कहलाएगा? पार्शियल एगोनिस्ट. यदि बेसल एक्टिविटी रिड्यूस हो रही है, then it will be called as inverse agonist. So these are the different forms of agonist. Okay, so drug molecules are prepared in this way only, that regulate the response of the receptor activity. Okay, नाउ लेट्स सी द मैकेनिज्म ऑफ एक्शन। तो अब हम देखने वाले हैं बहुत ही इंपॉर्टेंट टॉपिक जिसका नाम है मैकेनिज्म ऑफ एक्शन और मोड ऑफ एक्शन। अब न्यूक्लियर रिसेप्टर कैसे काम करता है ये समझिए। तो टाइप 1 न्यूक्लियर रिसेप्टर हमने देखा, बेसिकली जो स्टेरॉयड हार्मोन रिसेप्टर्स आते थे, प्रोजेस्टेरॉन रिसेप्टर, एस्ट्रोजन रिसेप्टर, ये कैसे एक्चुअल में काम करते हैं, कैसे जो है वो इनका मैकेनिज्म ऑफ एक्शन क्या है। सो लेट्स अंडरस्टैंड दिस। ओके, सो दिस इज द हार्मोन और वी कैन अंडरस्टैंड हार्मोन एज ligand. So basically ligand will cross the plasma membrane. So in the cytoplasm you can see this is the receptor. Okay, this is the nuclear receptor present, and you can see it is present within the heat shock protein. Okay, so these are heat shock proteins. These are basically chaperones. Okay, one example is HSP90, which is present in case of steroid hormone receptor. Okay, so in steroid hormone receptor, by binding to HSP, basically it remains in inactive state. So when the ligand binds, then heat shock protein is dissociated, and further the nuclear receptor and hormone or ligand complex forms. So the first step is ligand binding and detachment of the heat shock proteins, and after the detachment the nuclear receptor and hormone complex will form, and then dimerization of receptor hormone complex will occur. So you can see after the formation of nuclear hormone complex, nuclear receptor hormone complex, that dimerization occurs, and after dimerization it translocate. I have discussed earlier also that type 1 nuclear receptor needs translocation because its receptor is present in the cytoplasm. So it translocate by crossing the nuclear membrane through the nuclear pore, and further the recruitment of the co-activator occurs. So when the co-activator recruited, then RNA polymerase, further you can see transcription of the messenger RNA can take place, and further gene regulation occur. So the next step is translocation to the nucleus, further co-activator recruitment occur. So binding of the nuclear receptor dimer to the DNA response element, you can see this is the hormone response element where it binds, and further the gene regulation you can see occur. So mRNA, protein, and the physiological function can occur. Okay, so this is the type 2 nuclear receptor pathway. So type 2 nuclear receptor pathway का क्या क्या example था? THR. Okay, so retinoic acid receptors. So these are nuclear type 2 nuclear receptor. So in this case we have already discussed that these receptors are already present in the nucleus. So there is no need of translocation of the nuclear receptor. So basically the hormone or the ligand directly enters in the nucleus by crossing the nuclear membrane through nuclear pore. Okay, here you can see already these receptors are bound to the hormone response element of the target gene, but there is co-repressor which is already bound to the receptors. So co-repressor what it does, तो ये क्या करेगा, इसको इनएक्टिव स्टेट में रखेगा. इट मींस लाइगेंड की बाइंडिंग होगी तभी जाके ये एक्टिवेट होगा, नहीं तो ये बाइंड तो रहता है हार्मोन रिस्पॉन्स एलिमेंट से जीन पे, बट यहां पर को रिप्रेशर होने के कारण ये इनएक्टिव स्टेट में होता है. सो बेसिकली रिसेप्टर इज इनसाइड द न्यूक्लियस बाउंड ऑलरेडी टू द को रिप्रेशर, इट मींस दे रिमेन्स इन अ इनएक्टिव फॉर्म. तो यहां पर रिक्वायरमेंट क्या है? सबसे पहले क्या होगा, लाइगेंड की बाइंडिंग होगी टू द रिसेप्टर विच विल कॉज द डिसोसिएशन ऑफ द को रिप्रेशर. वहां पे डिसोसिएशन हीट शॉक प्रोटीन का हो रहा था टाइप 1 में, बट यहां पर को रिप्रेशर जो होगा वो डिसोसिएट करेगा, और जैसे ही को रिप्रेशर डिसोसिएट करेगा देन यहां पर रिक्रूटमेंट होगी को एक्टिवेटर की. तो यहां पर आप देख सकते हो को एक्टिवेटर का रिक्रूटमेंट हो रहा है, और को एक्टिवेटर का रिक्रूटमेंट होगा देन फर्दर आरएनए पॉलीमरेज विल बी असाइंड. So recruitment of the co-activator protein and recruitment of the protein for the transcription, and further you can see the gene regulation occur, and further protein will form and physiological function will occur. So this is the mechanism of action of type 2 nuclear receptor pathway. Now the intracellular receptors of the nuclear receptor. Now we talk about how much members are present in the nuclear receptor. So this nuclear receptor family tree has been shown here. Okay, so total 48 nuclear receptors are reported to be present in the humans. These all 48 nuclear receptors, as I have told in the first slide, that it can be classified into subfamilies based on the sequence homology 0 to 6. So you can see here nuclear receptors 0 to 6. 1, 2, 3, 4, 5, 6. So these are the subfamilies which contains different types of nuclear receptors, and there are total 48 nuclear receptor which forms a superfamily. Now these are some examples. These are endocrine receptors. I think everybody knows. These are glucocorticoid receptor. These are the ligands given in the second column. Mineralocorticoid receptor. Mineralocorticoid is the ligand. Progesterone receptor. This is progesterone is the ligand. एंड्रोजन रिसेप्टर एंड्रोजन उसका लाइगेंड होगा ER अल्फा बीटा 2 आइसोफॉर्म्स पाए जाते हैं ER के एस्ट्राडायोल और एस्ट्रोजन इज द बेसिकली ligand of the estrogen receptor, and these ligands are also termed as hormone. सो इंडोक्राइन रिसेप्टर टोटल कितने हैं 6 है. अडॉप्टेड ऑर्फन रिसेप्टर. ऑर्फन रिसेप्टर मैंने पहले बताया. ऑर्फन रिसेप्टर क्या होते हैं? सो बेसिकली ऑर्फन रिसेप्टर होते हैं जिनके लाइगेंड पहले क्या थे पता नहीं थे और बाद में वो लाइगेंड क्या हुए डिस्कवरी हुई. तो बाद में लाइगेंड जब पता चल गया तो उनको क्या बोल दिया गया अडॉप्टेड ऑर्फन रिसेप्टर. सो दीज आर सम एग्जांपल्स RXR अल्फा बीटा गामा. सो 9-cis retinoic acid is the ligand for the RXR alpha beta gamma. PPAR also comes under 3 isoforms, and fatty acids are the basically ligand. So it involved in the fatty acid metabolism. LXR, okay, farnesoid X receptor, pregnan X receptor. These are all orphan receptors. It means their ligands has not been discovered yet. अभी तक इसके लाइगेंड्स पता नहीं चल पाए हैं. एनिग्मेटिक ऑर्फन्स का क्या है? इसका भी सेम चक्कर एडॉप्टेड वाला, यानी कि यहां पर भी लाइगेंड पहले पता नहीं था, ठीक है, लेटर इसकी डिस्कवरी हुई. बट यहां पे इंपॉर्टेंट पॉइंट ये है कि इन्हें एनिग्मेटिक इसलिए कहा जाता, ये जो कॉन्स्टिट्यूटिव एंडोस्टेन रिसेप्टर कार हो गया, HNF हो गया, तो ये सारे रिसेप्टर्स के लाइगेंड तो डिस्कवर कर लिए गए, बट ये बिना लाइगेंड के भी काम करता है. दैट्स व्हाई दीज आर कॉल्ड एज एनिग्मेटिक ऑर्फन्स. नाउ जनरल फंक्शंस ऑफ द न्यूक्लियर रिसेप्टर. सी, nuclear receptors perform lots of functions. That's why they are very evolutionarily conserved. Okay, so it has a role in cellular differentiation, okay, development, cell fate. It determines the cell fate. It has a role in embryonic development. In metabolism, you can see PPAR gamma has a role in fatty acid metabolism. Glucocorticoid receptor is involved in glucose metabolism, so it regulates the energy balance, nutrient sensing. It has a role in disease pathogenesis. For example, breast cancer, you can see estrogen receptor is associated, androgen receptor is associated with the prostate cancer. Okay, now it regulates the gene expression, transcriptional activation, transcription repression depends on the recruitment of the co-activator and co-repressor, and hormone signaling that we already know, because it regulates lots of functions, okay, that we have already studied in endocrine system. Now let's move to the next slide, where I have given some you can say most popular examples of the nuclear receptor and their ligands and their functions. So quickly see these receptors. These androgen receptors, the ligand is testosterone and dihydrotestosterone. The function is it controls the male sexual development, muscle growth, and bone density. Glucocorticoid receptor, cortisol is the ligand. It regulates the metabolism and immune function. Progesterone receptor, progesterone is the ligand. It plays a crucial role in female reproductive processes, menstrual cycle, and also breast development. Now, mineralocorticoid receptor, aldosterone is the ligand. It regulates the electrolyte balance. Basically, it regulates the osmotic balance inside the body. Now, retinoid X receptor. This is basically a common heterodimeric partner for most of the nuclear receptor, and the ligand is 9-cis retinoic acid. Forms heterodimers with other nuclear receptors to regulate the gene expression. It has a role in growth, development, and vision. Now some more receptor. Estrogen receptor, I think everybody knows. Estradiol or estrogen is the ligand. It mediates the biological effects like female sexual development, reproduction, and bone health. Thyroid hormone receptor. Thyroid hormone like T3 is the ligand. It regulates the metabolism, growth, and development. Now retinoic acid receptor, the trans retinoic acid. The previous one was RXR. The ligand was 9-cis retinoic acid. But in case of RAR, Trans-retinoic acid is the ligand. It plays a crucial role in embryonic development, cell differentiation, and vision. Vitamin receptor. So 1 to 5 dihydroxy vitamin D3 is the ligand. It regulates calcium and phosphorus homeostasis. So it is very essential for the bone health and also regulate the immune function. Now PPAR alpha, beta, gamma, delta. So peroxisome proliferator activated receptor. So polyunsaturated fatty acid, lysophosphatidyllic acid, prostacyclin, these are the ligands. It regulates the fatty acid metabolism, placental implantation, adipogenesis, blastocyst implantation. It means it has a role in embryonic development also. So these are the some examples of the receptor. Okay, so these are the references that I have been used to prepare my slides. So thank you guys for your patient listening. So मुझे लगता है कि आप लोगों को ये टॉपिक समझ में आ गया होगा. सो प्लीज लाइक शेयर एंड सब्सक्राइब दिस चैनल. यदि आप चाहते हैं कि ऐसे ही टॉपिक्स पे हम लोग वीडियो बनाते रहें तो आप प्लीज कमेंट करिए. यदि आपको कोई भी डाउट होता है इस रिलेटेड तो आप कमेंट कर सकते हैं एंड वी विल ट्राई टू आंसर योर ऑल क्वेरीज एंड डाउट्स.